Okay, welcome to the Zarnescu Lab. This part of the lab is where the fruit fly genetics happens. We have a number of dissecting microscopes that people use to set up genetic crosses, to generate fruit flies with neurodegeneration. As an example here, Claire is an undergraduate student, and she is working on a project looking at insulin signaling in neurodegeneration. She's setting up some crosses, I believe. So you can see how she's putting the fruit flies to sleep, and then she's crossing the right flies to generate the right genotype. Here we have Sam, who is testing the effect of novel compounds with therapeutic potential in ALS. She's using Locomotor behavioral assay called larval turning to determine whether specific compounds that we have generated in collaboration with colleagues from chemistry can improve the locomotor function of our fruit fly models of amyotrophic lateral sclerosis. And she's actually identified a few completely novel compounds that we are planning to develop into therapeutic strategies with our colleague in chemistry and with help from Tech Launch Arizona, actually. These are additional fruit fly stations that people can use to do their fly work. They also can do dissections to isolate brains and study neurons in situ, in the actual tissue, under the confocal microscope, and look for various molecular markers of neurodegeneration. These are some benches. They are empty now because people are working in shifts or have class, where people generally sit and do desk work, if you want. Which is also very important, reading, researching. And if we can go into the other room from the lab, this is a lab. This is the part of the lab where we do biochemistry. We work with protein gels in order to look for various markers of neurodegeneration. So some of the techniques used in the lab are Western blots. Maria is an undergraduate student here who is making some solutions. She's planning to dissect some brains and look for a new target of neurodegeneration that we recently identified in the lab. And as we move forward, this is a bench where students will do Western blots. Once again, to do protein work, we use mass spectrometry as an approach to identify novel proteins that are dysregulated in neurodegeneration, with the idea that what we identify in the fruit fly, we can then validate in human tissues or patient-derived motor neurons. Other parts of the lab are, standard in any molecular biology lab. We have a hood, we have a bunch of heat blocks. This area here is used for working with compounds. We use fruit flies as an in vivo model to do what we call phenotypic screening and identify novel compounds or drugs that have neuroprotective potential. In other words, they can improve locomotor function and potentially increase lifespan in our models. Once we discover these, we then go ahead and test them in patient-derived motor neurons, and hopefully from there we can hope to one day move these into patients.